Hello students, in this video I'm going to focus on exercise 11 parts 2 through 4. I'm going to take a look at how you use the flylab simulator or the flylab simulation that we'll be using, and I will be following the exercise document. I have mine printed out right in front of me so I can see it and make comments on it as I go. I will be reading from that while sometimes looking at the flylab simulator, sometimes flipping back and forth between the worksheet document, the worksheet assignment, and the flylab simulator. So let's move to the worksheet assignment. Okay, and we have finished part 1 if you've done the first video, and now we're on to part 2. Part 2 is called the Monohybrid cross. And I see it's quite long, so I kind of scrolled through this, and I guess I'll be scrolling this way. We will have to do a cross. We will use the flylab simulator to do this cross. We're going to carry out the cross. We're going to answer the question, does the mutation that we're looking at appear to be dominant or recessive? We're going to do a second cross. And we're going to write down some genotypes and draw the Punnett square for the second cross. We're going to write down our predicted phenotypic ratio for cross 2 based on our Punnett square. Then we're going to go back and use the flylab simulator to analyze the data using a chi-square analysis and compare our predicted results from question 6 to our expected results. To our observed results, to observed results. And then we'll draw some conclusions. Okay, then we'll move into sex-linked traits and eye color using the flylab simulation. You'll see that the parts 3 and 4 are set up fairly similarly to part 1. So let's go back and take a look at the specifics of this part, the monohybrid cross. It says that we are going to use the flylab simulator, and we're going to study different traits depending on what your last name is. My last name is Jenkins, so I'm going to study ebony body color. It's not sex-linked. I know that already. I don't know if it's dominant or recessive. And I'm going to be making a cross between a female that has the mutation of ebony and a wild-type male. People who have last names of N through Z will be making the purple eye. You will be working with the purple eye trait, okay? So the first thing it says is write out the genotypes of the parent flies. Well, I don't know them yet exactly. I do know the wild type male, right? The wild type, so I'm going to write down the female. I don't know quite yet, but the male I know is wild type. So I'm going to put down plus because he has 2 wild type alleles. We know that she's going to be homozygote, but we don't know if ebony is dominant or recessive. So I'm going to leave that there. And then it says carry out the cross 1 in your flylab simulator and put your phenotypes for their offspring in table 2. Table 2 is a couple pages down on the worksheet, so I'm going to have to kind of spread out my worksheet so that I can do this effectively. And then it says, based on cross 1, does the mutation appear to be dominant or recessive? I guess I'm kind of waiting to find out if we get all offspring that have the mutation or if we get all wild-type offspring from the first cross. I'm going to have to do the cross before I move on. Then I'm going to set up a second cross, it says here. Okay, so I have to get to my flylab simulation. And this is kind of a fun little simulator. And in the flylab simulation, you're basically going to be using the crossing, this page, and the analyzing tab. So this page has a female or a male, and the females are bigger. They have less coloration on their abdomens, and they also don't have sex combs, which are these little bristles on the front legs of the male that help them mate. And our females can be have any number of traits, and so can our males. They start out as being wild type for all these different characteristics, okay? But I'm going to pick a female that has the mutation for ebony body color. That's what I'm going to do. So I need to click on body color and change her to have ebony body color. Ooh, that's beautiful. And I'm going to select her for mating. And there she is. She's got this big E. Doesn't mean that the trait is dominant. It's just their symbol that they use, okay? We don't know if the trait is dominant. And now I need to mate her with a wild type male. So I need to design my male. And he's going to be wild type, so he doesn't have any other interesting characteristics. He's going to be wild type. So I'm going to click on wild type. So now I've got my mating pair for cross 1. And I'm going to carry out cross 1 and look at the results, mate the flies. Now, initially, in these experiments, they report the flies with their sex as well. So here we've got the offspring of cross 1. I've got wild type females and wild type males. In other words, all the offspring are wild type. What does that tell us about the trait of ebony body color? It's recessive. Yeah. So that's going to help me with question 1. Write out the genotypes of my original parents. And it's going to help me with question 3. I found all wild type individuals. And now I have to report these results in table 2. So I have to write down 523 wild type females. Okay, and these numbers will be different every time you do them. You're going to have to do your own. And 511 wild type males. Okay, and I could, if I wanted to I could combine the two because they're all wild type and this isn't sex linked, but I'm not going to worry about that now. Okay, now I've answered questions 1, 2, and 3 for the monohybrid cross. And then it says, because I think this is recessive, because all of the offspring are wild type. I don't see any ebony body. And then I move on. After you've observed the offspring from cross 1, set up cross 2. Yay! For this cross, you will be mating a female offspring from cross 1 with a male from cross 1. Oh, we're going to be mating siblings. This is so easy to do, much easier than actually mating these. If you take genetics class later, you will actually be breeding fruit flies, and it's lots of fun. There are lots of fruit flies everywhere up on the 3rd floor. But we're going to mate the 2 offspring from cross 1. They're going to be mating siblings. I wonder what their genotypes are. You should know this, okay? In fact, you should write out the most likely genotypes for our flies. And we probably know this already. If I go back to the questions that we've been working with, I have to fill in this. My original parents were homozygotes, right? The female was ebony and the male was wild type. If they mate, what are all the offspring's genotypes going to be? They should be heterozygotes, right? 1 ebony allele and 1 wild type allele. So if I scroll down here, what are the likely genotypes of the F1 by F1 parent flies? I think they're going to be ebony, I think they're going to be like this. I think they're both going to be heterozygotes. They're both wild type in appearance, but they have that ebony allele. And then I can predict what the offspring from cross 2 will be that I'm going to mate in a minute on my flylab. So if I'm going to do the Punnett square for question number 5 for cross 2, I see that each individual, each parent, can either donate a little e or a wild type allele to the gamete, right? These are the possible gametes that the female can make, that the male can make, and I can fill in the possible results, the expected genotypic and therefore also phenotypic results. Da la la la. So I can predict what I'm going to see in this second cross. It looks like I'm going to see 1, 2, 3. 3 of the 4, oh, that makes sense. 3 of the 4 are wild type or expected to be wild type, and 1 of the 4 are expected to be ebony. So my expected phenotypic ratio, I'm just going to leave these in fractions. I like that so much better. 3 of the 4 offspring should be wild type, and 1 of the 4 offspring should be, in fact, ebony, right? La la. That's what my predictions are. Now I'm going to actually run cross number 2 and write down the results in table 2. Okay? You will need to use the Analyze tab on the flylab simulation to complete table 2. You will need to ignore the sex and include a test of the hypothesis. Okay, so we have to complete table 2. We've already added in the first row. I think I had 523 wild type females and 511 wild type males. You're going to have different numbers. Okay, and now we're going to run cross 2 and complete this table, and we're going to actually determine if our observed results in this simulation fit with our expected or predicted results that we just made up here in question 6, where we predicted a 3-to-1 ratio of this F1 by F1 cross. So let's go back to our fly lab and actually do the mating. Here are our flies to mate. I'd already selected them, right? The offspring from cross 1 times an offspring from cross 1. Mate the flies, and voila, here we go. These are the offspring. And I'm going to write them down into my box on second row of table 2. I'm going to write down 381 wild-type females and 402 wild-type males, 138 ebony females, and 106 ebony males. And then I'm going to do some analysis. At this point, I need to go to the Analyze button to complete the next rows of my table 2. And I've got all of my observations here. We counted 1,027 flies in this simulation. Super easy. Did it really fast just by the click of a button, right? I'm going to ignore the sex of the flies because this isn't sex-linked. You know, you can see that the same number of male and females, you know, virtually the same. Okay, we're going to ignore that. Ignore the sex of the flies. And I'm going to report here then the in row 3, the phenotypes of cross 2 offspring and their numbers of the flies. So I had wild-type flies, I had 783, and ebony flies, I have 244. I'm going to put those into my row 2, right? Okay. Now it says observed phenotypic ratio, predicted phenotypic ratio, predicted number of flies. How are we going to do this? Well, the observed phenotypic ratios are set up for us already. It looks like 76 or 0.76 of the offspring are wild type. So 0.76 are wild type and 0.24 are ebony. That's our observed phenotypic ratio, if you will, our proportions. If I include a test of the hypothesis, I have to insert what I expected to see. What percent of the offspring were expected to be wild type? Well, I have to go back to question 6, and I said 3/4 of the offspring. 3 divided by 4 is 0.75. 0.75. 75% of the offspring should be wild type, and 1 divided by 4, or 0.25 of the offspring, should be ebony. That's my predicted phenotypic ratio. 3/4 of the offspring, or 0.75, are expected to be wild type. 1/4 of the offspring, or 0.25, are expected to be ebony. Okay? And if I click test my hypothesis, then I should be able to calculate a chi-square analysis. Just like that, with the click of the button, we can look at, it tells us the predicted number of flies for each category or each phenotype. The predicted number of flies for cross 2 is already here. And I predicted 770 wild type flies. How many did we have? 783. That's pretty close. And we predicted 256 ebony flies, and we had 244. Okay, that's pretty close too. We calculate a chi-square, and our chi-square value is 0.84. That's our chi-square value. Our degrees of freedom are 1 because there's only 2 categories, and our level of significance is 0.35. What does that tell us? We're going to answer questions 8 and 9 on our worksheet. All right? What do your results tell you about the genetics of the trait you tested? Is the trait autosomal? Does it show a normal pattern of dominance and recessiveness? Yeah, it does. We saw that ebony seems to be recessive. It seems to fit with this we didn't see ebony at all in the first generation, and then it shows up in about a quarter of the population in the second generation. That's what we would expect if this ebony trait is recessive, right? And it's not sex-linked. There are no, there's nothing strange about the sex inheritance. So I'm going to say that it seems like it is autosomal and recessive. And do your data, did you obtain the ratios you predicted? Use the results of the chi-square analysis to draw your conclusions. Here, my chi-square value was 0.84, and my p-value, that's the level of significance, was 0.358. That's a big number. What that tells me is that it's likely that the differences we saw between what we observed and what we expected to observe are just due to chance. Yeah, that's great. Our observed data fit with what we predicted. Yes. And so we don't reject our hypothesis. Our observed data fit well with our predicted. And we know that because the p-value is so big. The p-value is bigger than 0.05. Anything bigger than 0.05, we can say that the observed data fit well with our expected data. We don't reject our hypothesis. So I'm pretty happy there. I'm going to fill that in as a conclusion in question 9 for our monohybrid cross. And then I'm ready to move on to our second cross. So we filled in this whole table. We filled in the chi-squared value, the degrees of freedom, the level of significance, or the p-value. We've answered the conclusion questions, and we're ready to move on. And we're going to move on to sex-linked traits. Now, in this part, you're going to take a look at one trait that is, in fact, sex-linked. It's white eyes. The mutation of white eyes is sex-linked. It's carried on the X chromosome. And you're going to make several crosses to kind of prove to yourself that this trait is different than autosomal. It's not on an autosome, but it's on an X chromosome, the gene for white eyes. So you're going to make several crosses with your flylab simulator. Okay, and then you're going to do some predictions. We're going to actually draw out the Punnett square for cross number 2 that you made. And I've helped you here by giving you the gametes. That helps a lot. You're going to think about what's the genotype of the female offspring, what's the genotype of the male offspring from that cross, and then you're going to make a third cross. And you're going to have to fill out the gametes yourself. And this is sex-linked, so you have to remember to use the X's and the Y's. And then you're going to run the cross. You're going to carry out the actual cross on a flylab simulator again, and do the chi-square analysis to compare your observed data from the simulation with what you predicted from your Punnett square up above. All right, you will not ignore the sex this time. So we're going to have to pay attention to the sex. If you have questions about this, I will be in my office hours, and I can help you during office hours. Okay? And then you have to draw a conclusion. Finally, we're going to do a dihybrid cross. That means we're going to study 2 genes at the same time, people. Whew. And if your last name starts with A through M, you're going to be studying sepia eyes and curved wings. If your last name starts between N and Z, you're going to study brown eyes and shaven bristles. All right, so in this case, you're going to set up 2 crosses again with your flylab simulator, and you're going to cross a female with both mutations times a wild type male. I want to think about this cross, so I'm going to give you some hints here for this dihybrid cross. I'm going to say that my last name is Rosema. That's my maiden name. And so I'm going to look at brown eyes and shaven bristles. Okay? And I am going to mate a brown-eyed, shaven-bristled female with a wild-type male for the first cross. Right? That's what it says I need to do. And so what are the genotypes of my parents? Well, I don't know if these traits are dominant or recessive. So I'm going to think about this for a minute. I'm going to write down the female. The male is super easy. The male is wild type for both body color and for bristle shape. Okay? So he's going to be plus. Ooh, that's, he's going to be wild type for for the eye color and wild type for the bristle shape. And I'm always going to write them in the same order because I can't tell one plus from another otherwise, right? So I'm always going to put body color first. The female, I don't know if these mutations are dominant or recessive, so I don't know if I want to use capitals, but what if they're both recessive, then I would say she's brown and shaven or something, and shaven, something like that, if they're both recessive. But I don't know that yet. I have to run the cross, all right? And then I have to determine in question 2 if the traits are dominant or recessive based on the results of that cross. If they're both recessive, we expect to see all wild type males, all wild type individuals in the second generation, right? So let's take a look. I'm going to run a new simulation. No, I don't want to go here. I want to go to design. I'm not sure if I don't know how exactly to reset these. This doesn't seem to reset, but I'm going to try it. My female is going to be brown-eyed with shaven bristles. So what her eye color is brown. This is funky, isn't it? Look at those crazy eyes. And her bristle shape, where is that? Bristles. Well, look at all those different bristles you can have. I'm going to go with shaven. So she's gorgeous, gorgeous. We're going to select her for mating. And we're going to mate her with a wild type male. That's easy. I'm going to design my male fly, and I'm going to design just the wild type fly. Oh, there they are. They're lovely. They use these symbols for shaven and brown. Don't worry about them. I'm just using B's and S's. Makes it a little easier. I'm going to mate my flies. Ta-da! It says cross 3 because I somehow didn't reset my crosses. Somehow I have to do that, but don't panic. This is my first cross for the Dihybrid cross. And it says here, based on your first cross, identify whether each trait seems to be dominant or recessive. And so I'm going to look at this and I'm going to go, oh, all the offspring in cross 3 are wild type. All the females are wild type, all the males are wild type. So what does that tell me about brown and shaven bristles? I think both of them are recessive, right? Because brown eyes don't show up and neither do shaven bristles. Okay, that was easy enough. Then it says, after you observe the results of that cross the offspring from cross 1 with each other. Ooh, another sibling mating. This is exciting. And then for cross 2, write out the genotypes of the parent flies. This is question 3. I'm going to go to that. I think I can do that. Okay, so I actually correctly suggested the genotypes for the parents of the first cross, and I think that both traits are recessive because they didn't show up in the first generation, right? Period. Okay, now I'm going to make the cross of these individuals that have inherited the first generation individuals, or the first filial generation individuals, inherit one brown allele from mom and a wild type eye color from dad, one shaven bristle allele from mom, and a wild type bristle thing from dad. So I think the genotypes of both the parents would be like this: brown with a wild type, and shaven. Not that. I'm doing some bad things here, people. I'm making trouble. Okay, and shaven with a wild type. Oh, wow, am I having trouble? Okay, I can blame that on my thumb. That's going to be the parent. That's going to be both parents. Brown. They each are heterozygous for each trait, right? Okay, I'm ready to make a Punnett square. Draw a Punnett square for cross number 2. Oh my goodness, this is going to be scary because we've got 2 genes. And we've got 2 parents that are heterozygote for both genes. So if I'm looking at the first parent, you need to have both. You need to have a copy of the eye color gene and one copy of the bristle gene in each gamete. So I'm going to put I'm going to always put the eye color gene first, B, and the bristle gene. So you could get BS. Yeah, that's what this is, BS. You could get a little B for the eye color and the wild type allele for the shaven bristles, or for the bristle shape. You could get the wild type allele for the eye color, and you could get shaven for the bristle type, or you could get wild type for the eye color and wild type for the bristle shape. Whew! So we have one copy of each gene in each gamete. We have one eye color and one bristle shape. 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 And I'm going to have to put all of these down here too. Okay, so you can fill that in. Then you have to start sorting it out. So I'm just going to do this. Okay, then you have to fill in the possible offspring, right? B, B, S, S. That's one of them. Okay, and then this would be B, B, oopsie, whoa, wait, why does it do this? I'm going nuts out here. B, B, S, positive, okay? And then the next one, B, S, S, okay? And then B positive, S, B positive, people. I gotta be positive, this takes forever. I'm not good at doing these. Look at this mess I'm making. Okay, there we go. And you're going to fill in all 16 of these boxes. And then you're going to answer the question. Record the expected phenotypic ratio from this F1 by F1 cross here. Did your punnet give you a predicted 9:3 to 3:1 ratio? Oh yes! This is a dihybrid cross, where the parents are both heterozygous for both traits. We should see a 9:3 to 3:1 ratio. Oh, but don't just write 9:3 to 3:1 ratio. Write 9/16 of the offspring should be, 9 of those 16 boxes should inherit, I think, both the wild type alleles, like both the wild type traits. So should be wild type for eye color and wild type for bristle shape. So 9/16 should be wild type for eye and wild type for, I'm going to count them all up, but I'm going to, for bristle, okay? And then 3 of the 16 boxes should be wild type for eye, but also, and shaven for the bristles. And 3 of the 16 boxes should be the opposite. Should be brown-eyed and normal bristles, wild bristle. And 1 of the 16 boxes better be brown-eyed and shaven bristles. I think that should be it. I should see that 9:3 to 3:1 ratio. And it's only if I've done it correctly. If you haven't filled this out correctly, you're going to be in trouble. But you're going to have to sort out each box to make sure that you get that 9:3 to 3:1 ratio. Then you're ready to actually run the simulation, run this cross, and compare your results to the results of, to our expected 9/16 to 3/16 to 3/16 to 1/16 ratio. And you'll see if the data fit well with our prediction. All right? So that's what you're doing in this last part of the assignment. You're going to have to go back and you're going to have to mate your offspring. And it's super easy to do, right? So you simply mate those two and you get a bunch of offspring. Oh, look at all the offspring. We can go to the Analyze tab and ignore the sex of the flies to compare our offspring there, okay? That's going to be how you do the last component of this exercise. And I think I'm tired. So if you have questions about this, come to my office hours and visit me. I will enjoy seeing you at them. That's it. Thanks.